Okay. Bueno, bueno. Sí, prueba, prueba. Bueno, bueno.
Si quieres hablar acá en uno.
Bueno, bueno, probando, probando, probando. Bueno, 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 probando.
Sorry, okay, there was a problem with my name. Okay. Good morning, everybody. Please join this meeting. Hi, good morning. How was your night? Perfect. Welcome once again. Let me check if everything is okay with the broadcast. Guys? David? Everything is okay? No, okay. We need to wait a bit more. Thanks once again. So it's a pleasure to introduce today to Dr. Catherine Stanger Hall, also a legend, the same as Sarah Lewis and Juan Yuso just in yesterday's plenary talk. Dr. Catherine Stanger Hall is professor at the University of Georgia, Athens, in USA. After earning her PhD at the University of Tübingen, in Germany, she taught at Duke University, Wake Forest University, and the University of Texas at Austin before joining the University of Georgia. In her PhD, she used molecular phylogeny to study the evolution of fireflies, their communication signals, and their sensor morphology. Please welcome Dr. Catherine with this plenary talk with the name An Updated Worldwide Phylogeny of Lampyridae, New Insights and Next Steps. Thank you.
Good morning, everyone. Thank you, Sistel, for that introduction. And of course, I want to thank Finn, our hosts, and all the organizers for making this symposium possible. And I also want to thank all my collaborators and our students that contributed to the work in this talk. And let me just do this real quick. And this includes, see how that works. Oh, next slide, please. This includes Gavin Martin, who led the charge on the first worldwide firefly phylogeny. Then Gareth Powell dated this phylogeny. And then Yelena took the lead on generating the second version of the worldwide phylogeny. And it's most of her work that I will be talking about today. Now, all this work was generated in close collaboration with my collaborators, Seth Bybee and Mark Branham, and Luis Silveira as a postdoc on the project. Our current project is the Lucidoda project with Luis and Joe McHugh as co-PIs. And this will include the third version of the worldwide phylogeny. Now, I want to thank my many collaborators around the world, or our collaborators around the world, without whom none of this would be possible, who generously contributed specimens for our studies. That's going the wrong way. Sorry, no. Anyway. That's not quite it, but anyway. So thank you to all of our collaborators. And I want to acknowledge here that we all stand on the shoulders of giants. And for us, these are Jim Lloyd and Leslie Ballantyne. Leslie has been helping us putting the correct names on our Asian and Australo-Pacific fireflies. So thank you for that. Now, fireflies are very popular among citizens around the world. And we can see that on this map from iNaturalist, where everybody can post their observations and photographs. And this was a map generated by the Finn group that was on iNaturalist that was started by Tim Fallon. Now, all these points here are generated through 200,000 individual observations. And this is how they break down in different regions of the world. Now, here listed here, about 500 different species, and assuming those are correctly identified, this makes up about 20% of the population of the species that we know are present in fireflies. As of last week, Oliver Keller's Lampyridae of the World had a count of 2,625 described species. All right. So all these species, they vary greatly in their morphology, as you can see here, in their habitats and in their behaviors. And so how do we make sense out of this diversity And how can we study the evolution of firefly traits and which traits are important for taxonomy? So this is where where phylogenies come in. Phylogenies are an important organizing principle for diversity, and Charles Darwin was actually first to conceptualize the relationships of species to think about diversity and evolution. Ernst Haeckel was one of the first scientists that then basically generated big trees with lots of different organisms. And since then, of course, there were major breakthroughs in phylogenetics and taxonomy that led us to taxa now based on shared ancestry and shared derived traits. And the importance of phylogenies for fireflies is illustrated by an increasing trend in publications that use phylogenies. And then most of these publications also discuss trait evolution. So this lets us identify convergent evolution, independent events of evolution, and how natural selection or sexual selection may have shaped them. And this is, let me go to the next one here. Let's see what I can do to mine. Sorry, my technology does not synchronize. And of course, this now lets us to study more and more evolutionary traits in fireflies around the world for more and more taxa. So what I want to do today is give you an update on our worldwide phylogenies and also what we have planned for the next 4 years. These are covered here by the first 3 points of this overview. And then I want to give you some example that directly emerged from our phylogenies, how phylogenies can actually help us answer important questions about firefly evolution. Now, these are topics that we are very interested in, but you could do that for any traits that are close to your heart. For the origin of firefly diversity, we actually have to travel back in time for about 41 million years. This is how the world looked like at this time. This is when titanosaurs roamed Argentina, Antarctica had an almost tropical climate, and this is when the first fireflies evolved. Now, we don't know exactly how those first fireflies looked like, but we know they had bioluminescent larvae and non-bioluminescent adults. McDermott has described South America as the cradle of firefly evolution. And so South America seems like a good bet for the origin of fireflies, but only big phylogeny with dated fossils will tell us that answer. So fast forward to today. Today, we have more than 2,600 described species in 12 subfamilies. And of course, species are added continuously to that species count. Now, 8 of these subfamilies were included in the first version of the worldwide phylogeny that was published in 2019, including the subfamily Lamprohicinae, which was erected in the study. If you want to hear more about the defining characteristics of Lamprohicinae, please see Alona Urkel's talk a little bit later today. Now, after Gavin published his study, it was Yelena Pacheco's turn, and she extended the taxon sampling to a total of 339 species in 91 genera in 11 subfamilies. This means that 11 of the current the 12 subfamilies are included in her phylogeny. The new subfamilies are Cladodynae and Chesprotainae that are represented for the first time in such a huge analysis. We also have a representative of Chegovarinae. However, we are presently still placing that on the phylogeny, so I can tell you more about that in a few weeks. Yelena's taxon sampling also included many more diurnal taxa and, of course, more neotropical taxa, which is the possible origin of firefly diversity. Just to give you an idea how these phylogenies are generated, we basically extract DNA, sequence the DNA with a probe set of 560 loci, and then Yelena combined her sequences with those of Gavin for a total of 397 taxa, 516 loci. But you can imagine if you have such diverse taxa, some of them will sequence better than others. So to reduce the missing data in our analysis, we eliminated some of the taxa which bad sequence result with the sequencing results, and also some bases that were very fully represented across all the taxa. So our final phylogeny included 342 taxa and 458 loci. Now, if you look at the resulting phylogeny and compare version 1 here with version 2, you immediately see the effect of taxon sampling, right? Yelena included 2 more subfamilies, and in addition, many more taxa across all the subfamilies. And so as a result, for example, if you look at it, her phylogeny included 16 taxa here for Autotetrine versus 2 taxa for the first version. And so as a result of that increased taxon sampling, we get a reshuffling of relationships. For example, let me go a little bit more in detail here. The Autotetrine are now at the basal group of fireflies, the ones that first diverged from the common ancestor of fireflies here. And what you notice here is, well, I don't know whether you notice it from the pictures, but these are non-bioluminescent adults, mostly. They tend to have short pronotomes, which is different from most other fireflies. And you also notice they're actually grouping in 2 different groups. They're paraphyletic, which means that this group includes their common ancestor here, but not all the descendants, because that's actually Lamperidae, right? So this is, of course, a taxonomic problem, and that means Autotetrine have to be split into two subfamilies. Now, this confirms what others have found. For example, Yang in his morphological study, and Yang and co-workers with some mitogenomes for some of the species as well. If you move down here, Cladodine here, that was erected based on morphology in 2022, you can see here was confirmed by molecular data to be an independent subfamily. And then Chesproetine was included for the first time and now grouped as a close relative of Polyclassis. Now, Chesproetine are really surprising, and you can see that when you look at their morphology, right? They look anything but a firefly. So these were confirmed now actually be members of the family Lamperidae, and specifically cyst or closely related to Polyclassis. Now, Polyclassis has been shuffled around quite a bit among different subfamilies. Right now, it's with Chesproetine. Now, there seems to be some evidence that, based on morphology, that maybe Polyclassis is actually a Nearctic species of Polyclassis bifaria, is a Nearctic species of the subfamily Siphonocerinae. Okay. Right. So if this would be the case, we actually would have all 12 subfamilies waiting placement for Seguaria represented in our second version of the phylogeny. However, we will have to include more Cyphonosorine taxa on the phylogeny to actually test and verify that, among other things. Now, aside from placing subfamilies more confidently now, we also made some headway on difficult genera. And so you may remember Vesta. Vesta is a genus that is represented in Asia and in the neotropics. And there was a debate long ago whether they should be split because they're so far away or whether they actually belong to the same genus. The news here is they belong together. And they are a relatively derived member of the Photorini here. Okay. In addition, another example is Lucidoda. And most taxonomists are aware that for Lucidoda is a mess, right? Because taxonomically, any species that do not fit in any other group are put in Lucidoda. And you can see that clearly on this morphological phylogeny from the Silvera lab that basically is largely consistent with our molecular phylogeny. If you look at those red names here, these are all Lucidoda, and they group with very different genera. Okay, so clearly what we need to do is sort out Lucidoda to make sure, to maintain the integrity of Lucidoda, and that will also impact the integrity of Lampyridae as a whole. And this is also where the Lucidoda project comes in that we just started a year ago. Okay, so this project has focused on South America. We will sample taxa across all biogeographic regions, thanks to our collaborators in South America that provide their Malay strep or their other firefly samples for this study. And then we also will focus in on Lucidoda and related genera to delineate Lucidoda as a clear taxon, as a valid taxon. We will present all these data or upload them also on a TaxonWorks data hub to make them available to everybody. And one of the big goals of our project is to train the next generation of integrative taxonomists. And I guess we got the message yesterday that this is an important thing to do. So let me introduce the Lucidoda project team. Okay, so this is Louis and Joe. These are our morphology specialists. My focus is more on molecular phylogenomics and on evolutionary trait analysis. Our students, the next generation of systematists that hopefully will out-expert us all in these areas in the not too distant future, are Julie Cadigan here and Michaela Humphrey from Western Carolina University, Martha Drake and Kimberly Garcia at UGA, and right now arriving in Athens, Georgia, are Andrea Cubillo and Miriam Romero. Some of you may know. In addition, what I want to say, you've heard Julie's talk last night on miniature fireflies. Please also check out Michaela's, Martha, and Kimberly's posters during the poster session. All right. Now, again, we have many collaborators on the project without whom that would not be possible to name individually, but I want to just mention Oliver Keller, who created the Lampyridae of the World database, and he is now transitioning the TaxonWorks data hub to a community governed resource. And so what we want to do is to, again, put our data on TaxonWorks and then connect that to Oliver's catalog, which will allow us to interconnect everything and expand this great resource. Right. So clearly, phylogenies are key for taxonomy. And I want to illustrate now with 3 examples how important they are also for evolutionary studies. Okay. These, as I mentioned, these examples directly emerged from our research interest and from our worldwide phylogenies. And I'm going to be using the full phylogenies for the first two questions, and then a smaller scale, just a selection of taxa from that phylogeny to talk about the evolution of potential pheromone sensor and what that means for pheromone use of fleshing taxa. So starting with bioluminescence, okay, why did adult bioluminescent originate? So we know from Gareth's analysis of Gavin's phylogeny that Lamperidae originated in the early Cretaceous about 141 million years ago. And then it took about 8 million years for the first bioluminescent adults to show up. Okay, so why? Why did they all of a sudden start to light up? Well, we think either it could be as a sexual signal. It originated as a sexual signal, facilitating mate recognition during mate search, or it could have originated as an aposematic signal, just like in Lowry, to advertise defensive toxin, the lucibufagins, to potential predators. Now, if it originated as an aposematic signal, then the relevant predators that were the selective agents for those signals should have been present, right? So when Gareth did this analysis, what we found was, again, first adult bioluminescent was about 130 million years ago. If you look at the potential predators that could interact with flying fireflies, they actually evolved much later. So no, they could not have been the selective agent to select for the first origin of light. However, once those light signals were there and used by fireflies, bats could use the light signals just like the wing beat frequencies of those insects and their zonal signatures to help them to avoid firefly faster, which may have given them some benefit as well. Now, something what is really fascinating about any organisms, not just fireflies, that use light as a sexual signal, in all those lineages, we see that there is a species proliferation once they use bioluminescent signals as mating signals, compared if you look at their sister lineages that don't use light signals. And this proliferation of taxa, of course, means also proliferation of different signals so they can distinguish each other and find a conspecific mate. So in fireflies, this means we see an amazing diversity of light signals. We see close, like here the blue ghosts. We see short flashes that are emitted in species-specific flash patterns. Those flash patterns can be synchronized or not. And also different species of fireflies can vary in their light color. So there's amazing diversity in their light signals today. What I want to focus on for today is look at with our phylogeny, we asked how often did glows evolve? How often did flashes evolve as a basis for studying those traits? So just as a brief reminder, how we study or trace or determine how many times we have independent evolution of traits in a phylogeny, I'm going to use the species phylogeny with 6 species, and they have 2 character states, either black or green. And in this scenario here, using parsimony, so the least amount of change required as an example, this species could be black because the common ancestor was black, and so are these 2. And then but that requires, to explain this pattern in present species, that there was an independent gain in this lineage, independent gain of green in this, and again here. So the minimum amount of changes are 3 changes, which are all gains. Okay. However, just because that fits doesn't mean it's true, right? There's an alternative way to look at this, because again, the transition to green here, the gain of green could have happened in that common ancestor, which makes those two species green. Now this requires a loss here of green, which makes those two species black. And then to explain this, we have another gain. Okay. Now these are equally parsimonious because they have 3 changes. But this one has 3 gains. This one has 2 gains, right? And a loss. And this is the reason why you will see a range of numbers when we talk about independent gains of different traits. So how often did cloning signals evolve? Well, Jelena used the literature of about 227 species total that use different mating signals and plotted them, looked at the phylogeny, and then we found that pheromones change to close 2 times, flashes to close 3 times. And then because we don't have data for all the taxa, an unknown ancestor state changed to close as well. So here's an example how a flashing ancestor changed to close in Micronaspis gabrielle here. Now, how about flashes? Okay. For flashes, we have, depending on how you count it, 5 to 9 independent origins of flashes on our phylogeny, most from pheromones. However, one was the switch was from close to flashes, like shown here in this example. Now you notice some of those taxa have no squares. Sorry, I'm not sure what happened. Can you pull up the presentation again, please? All right. Okay, okay. If you look here, thank you. If you look here, some of those, we have the character states here from the literature, but you notice some taxa don't have those data there because we don't have data from all the taxa on our phylogeny. Which means we will need the light signal data for all these species, but fieldwork can be difficult, right? And those signaling data are hard to come by. So wouldn't it be great if we could look at museum specimens and the species we have already in collections to infer their light signal type? So we looked at the light organs of different species. Let's see. Next slide, please. Can I? Great, thank you. So we looked at the light organs of 200 species with intact light organs, and we categorized them based on 5 of the shapes described by Brenham and Wenzel. And you can see some light organs cover the entire ventrites. Others have reduced sizes and are represented by bars, by single dots, by paired dots, or in such a split shape. And if you look at those red numbers, you can see how often they evolved independently on our phylogeny. I think these are astonishing numbers, and that just means light organ shape in fireflies is extremely labile and can change really quickly, even within groups of fireflies. So next slide, please. So if you look at Luciolini, what we can see is that we, from a likely ancestor, if you look at all this turquoise here, that covered both dendrites, we have now shifts to different shapes here, like paired dots in this group here. We have split light organ shapes here, here, and here. We have paired bars down here, and again, I think one's up there somewhere. So again, great variation in those groups. If we look at Lamparine, same thing happening. We have like 4 different light organ shapes represented in today's taxa, many of whom have those covered ventrites. This is same is true, by the way, for Torine as well. So obviously this is very labile, but so how can we use that to determine whether there's some correlation with signal type? So what we decided to do, obviously only those completely covered ventrites have the full possible size. These are all represent reductions in size. So we decided to measure the light organ area of these to basically to test whether the signal type could impose selection on light organ area, which means basically answering the question, do flashing species have bigger or smaller light organs than glowing species? Now, signal type is not the only influence on light organ size. Body size can play a role. And of course, phylogeny. Related species have similar traits. And so what you see in today's species could simply be inherited from the common ancestor and not to selection. So we can test for the influence of phylogeny using a phylogenetic signal that's Pagel's lambda, okay? And we did this both for absolute light organ area, which is simply the size, and relative light organ area, which is basically the light organ area relative to how big it could be, right? This is the kind of representing the reduced shapes. So this is the result, okay? Relative light organ area has a strong phylogenetic signal. So the shape is inherited basically completely from the common ancestor. If this is 1, it's fully explained by phylogeny. So it's basically 1, right? However, if you look at absolute light organ area, the size, it's so small, phylogeny has no influence on that. So we decided to test this with PGLS, phylogenetic generalized least squares, that accounts for phylogeny. It takes that branch lengths in as covariance of the data. And then we tested the model that the absolute light organ area is basically a result or influenced by signal type and body size, okay? So I have to tell you, light organ size is not a predictor of bioluminescent signal type, even though it's not influenced by phylogeny. Body size seems to be the major dominant influence here. So at least we can't use it on its own. So unfortunately, that means we still have to go out in the field and collect the data. We can't just use museum collections to infer signal type. And so what I would like to suggest is basically use extended phenotype labels whenever fireflies are collected, where the signal type and also flash patterns information is noted for the different fireflies. So we can utilize this for evolutionary study. And if you have any information on new species, what the signal types of your species are, please let us know. We would appreciate that. Now, so far, we've been talking about light signals, right? But as a matter of fact, diurnal fireflies do not use light signals. Instead, sedentary females, in most cases, will release a pheromone plume, and the male navigates through that plume to find the location of a conspecific female. And you saw a beautiful example of that yesterday in Photinus carrascus that Sarah Lower talked about. So, okay. When we look at our phylogeny, we find that pheromone changed twice to glows. They changed 5 to 8 times to flashes. However, stunningly, I think, 18 to 23 events of losing bioluminescence and going back to pheromone use, okay? Now, how is that possible? Does this mean species that go back to using pheromones here, do they reinvent pheromones and the sensors that they need to detect them? Do they keep the machinery and simply switch it on and back off, right? Depending on whether they have bioluminescence or not. Or is there a possibility that all species still use pheromones, release and detect pheromones to a small degree, depending on the relevance, how important it is in their habitat? Okay, now we know that glowing species also use pheromones from the work of Raphael Decock and Martin Nowak. So the question is, do flashing species too? Okay. Now, to get at this question, we can think about flashing species. Well, why would they want to use pheromones, right? Well, if you have watched fireflies in the field, aside from those whole trees lighting up, right? The light reach of individual fireflies is relatively limited. So it would be possible that even lighted firefly would benefit from pheromones to draw males into the area where females are and could respond to their signals. Okay. So we clearly need many more pheromone studies and behavioral studies in the field of glowing and flashing species to do that. But in the meantime, we have some morphological evidence. For example, we know that antenna size differs between diurnal species and nocturnal species. So diurnal species have a much, much larger antennal surface area. So the general assumption is that they have more chemosensors, more chemosensilla on those larger antennae. But do they, right? So do larger antennae have more sensilla in fireflies to increase their sensitivity to pheromones? Now, if you think about sensitivity, you could imagine that increasing antenna size and having more sensilla is not the only way to do it. You could also keep smaller antenna, just increase the density, or you could do something in between. So how do fireflies do it? Yelena answered that question by studying the antennal sensilla of these 7 species. These are not diurnal species, 1 glowing species, and then 3 flashers. She did that by looking at their individual antenna with scanning electron microscopy. She took more than 2,000 images, counting more than 140,000 sensilla, each separated by morphotype. And then she measured the total antennal area and also body size of the different specimen. So what did she find? Sorry, here we go. She identified 14 sensilla morphotypes. 10 of these are new for fireflies and 2 are new to beetles. Now, among these are 9 chemosensilla, which one of these could potentially, or several could be potential pheromone sensors. Now, if you looked at antenna size, yes, indeed, larger antenna have more sensilla, and you see that's true for mechanosensilla as well as for chemosensilla, and this is the sum of all of these together. Surprisingly, to me, larger antenna actually have a lower density than smaller antenna, okay? So you see some fireflies pack them more tightly. Now, what this means is for most species, antennal sensitivity is increased through selection for larger antenna, which means more sensilla. But then we also see some species like these Faustus males here, that if species have very small antenna, they're actually packing them in for higher sensitivity. All right. Now, if we look at the individual sensilla morphotypes and how they're distributed, so the sensilla types here, distributed against species, what you can see here, that the distribution is pretty consistent across mechanosensilla. Almost everybody has all the sensilla types. But in terms of chemosensilla, they're extremely diverse, right? Now, again, Sarah Lower, so far, the only species for which we know they have pheromones and we identified pheromones and Sensilla now is Carascus. But what about all the others? So again, we didn't want to wait for studies with pheromones. So what we did, we used morphology as a predictor and expecting, since pheromones Sensilla are one chemosensilla type, so chemosensilla should be more abundant, right, in males than in females, and this is indeed the case. Okay, compared to mechanosensilla, where there's no difference between the sexes, males have significantly more chemosensilla than their female. Now, in this case, these are the chemosensilla of both nocturnal and diurnal species together. If we look at them separately, what we see is there's a significant difference in males have significantly more chemosensilla than females. The trend is there, right, in nocturnal species, but it's not significant. And so we don't know right now whether we need more species, that's a power issue, or whether numbers are not the whole story, because we know that Phausis, we don't have data for that specific species, but we know that Phausis reticulata, that's something Raphael de Kock has shown, is that they're actually using pheromones. So is there a way that we can identify pheromones in Sensilla? Just looking at our collection of 9 different chemosensilla type, can we narrow that down? And again, using the criteria that a pheromone sensillum has to have pores to get the volatile chemicals inside where they can bind to key olfactory receptors. And also that, again, it has to be, that individual sensilla type has to be more abundant in males than in females. And using this, Sarah identified the potential pheromone sensilla. Again, we know this is no longer potential, this is verified. And it's the B1 sensillum specifically that Yelena identified as the one that responded to Sarah's pheromones. Lucidoda, another type, Purapica, another type. Phausis, the glowing species, T1, which is their only chemosensillum. And then Photinus spiralis, the bioluminescent close relative of Photinus carascus, also have B1 sensilla, and you see the sexual difference, the sex dimorphism here in sensilla numbers. Okay, now if we look at that on the phylogeny, this is how it looks out. So again, these are representatives of 4 different subfamilies, and I'm including Nippona cruciata here. These are literature data from Iwasaki from '96. And what you see here, B7 is present in almost all fireflies except for Phausis, which lost it. So this is an ancestral sensillum. It's not a pheromone sensillum because it occurs in very low numbers. And morphologically, it looks like it could be a hygrosensillum that senses humidity, which makes sense for soft-bodied beetles like fireflies. Now, overall, you see there's a lot of gains and losses, right? On this phylogeny, but I want to point your attention here again to close relatives Photinus carascus and Photinus puralis, with Photinus puralis being bioluminescent, Photinus carascus recently reverting to pheromone use and giving up light signals. So we know that B1 is the pheromone sensillum for Photinus carascus. Now, if we use our predictions, what we see is here in red, our pheromone sensilla candidates. Now, remember, these are candidates. Those need to be verified by functional studies. However, I think this gives us a good start of where to look first to see whether these could be pheromone sensilla in those different groups. Okay. So if we talk about bioluminescence versus pheromones, Do bioluminescent species use pheromones? Well, we have evidence from glowing species. And it seems now, in addition to the pheromone, the olfactory sensors that Sarah Lower detected in Phytinus puralis, I think there's accumulating evidence that some flashes may indeed as well. Now, whether they do or not, and it may differ between flashing species depending on their history and on their habitat, but there is some evidence that some of them as well may be able to do it. But clearly we need to test all these pheromones. We need more field studies on glowing and flashing fireflies and to actually test that idea. But I like the idea because it would explain why fireflies can just switch back to pheromones so many times on the phylogeny. So where do we go from here? Okay. Our project that I introduced, when we look at the worldwide phylogenies, the Lucidota project now is focusing on taxon sampling in South America. But you may notice a big area of need is Africa, right? And I hope that some of you will start working on Africa. And so we invite anyone to use our angled hybrid enrichment probes for your study taxa, wherever they come from. So to integrate them into worldwide phylogeny and see where they overall fit. Or of course, you can use the existing phylogenies to study any traits that you are interested in and that are close to your heart. In any case, we would love to talk to you, and I'm happy to answer any questions. I think I'm in time. Okay, thank you.
Oh, thank you. Yeah, hi. I came in late, but I've seen fireflies with, let me give you an analogy, a sheet with one light bulb behind it in the center, so that the light is stronger in the center than it is to the left and to the right of it, which means the reflecting layer is picking it up just like frosted glass would. So you're looking at the frosted glass, but you're not looking at where the actual organ, by dissecting where the organ is, how big it is, right?
No, we basically looked, if you look at the light organs, you really have that kind of ivory, white, yellowish structure on those dendrites. And we basically looked at how externally not lit up the size was of that light organ. Yes, definitely.
Understandable.
Yes, and so Mark Branham's group has started to do some CT imaging of light organs of fireflies. They've looked at the tracheal system supporting fireflies. I don't know how closely they looked at the surface itself, but there's still one paper waiting to come out that compares different.
I've seen that work, and it's inferior to electron microscopy. Anyway, my advice is to go, and I'd be glad to, I've got it all figured out, I'd be glad to consult on that.
Great.
But the electron microscopy is by far more efficient and precise,
Yes.
and ultrastructure clear down to the cells. Okay.
But I think it was an excellent start, right?
Oh, absolutely.
Get at it.
Absolutely.
And so there's always ways we can improve. And so ultimately, that's a question of having resources, right? To support
Yeah,
students.
absolutely.
And
Yeah.
I appreciate your willingness to advise.
And I have another one, but let's go to.
Yeah.
Hello, Catherine. Thank you for your wonderful talk.
Thank you.
I have a little concern in the approach. So you are in the phylogeny of sexual communication. You have pheromones, which is clear. Those are used for the communication between males and females. But with the bioluminescence, I think there is a kind of a mix of functions.
Yeah.
Sometimes it's for sexual communication, but even flashes might in some species only be used for aposematism.
That's right.
So that information should maybe be included
Yeah.
into the
So,
system.
yes, excellent point. And this is something, so that's why we're just talking about where the light signals are present versus not to see whether they can make two different signals. Clearly, ideally, we want to differentiate to what degree it's mating signal, what degree aposematism. But as you pointed out, we need many more data, and all species, even flashing species where it's a clear mating signal, can use it when they're caught in spider webs or if you catch them by hand. So we don't necessarily say that close in any case have to be a mating signal, but there can be used both close. Those species can use both closed
Yeah,
and
and
flashes.
even flashes, we think now from
Okay.
European Luciola species, we think the males and females don't have communication, but the males just see a different kind of flash, recognize it as a female, while the females don't respond to the males. So why do the males flash? Is it to warn off predators like bats? That's
Yes,
kind of a
I mean,
hypothesis, but
it's-
sometimes even flashes might not, because we always think that flashing species, by definition, use flashes only or to the side of aposematism for sexual communication, but flashing species might only use it for defense. That's a little point I want to make.
Yeah, no, excellent points. And I want to emphasize that especially we know a lot about North American species because of Jim Lloyd. So we are very familiar with what they do. But like you mentioned, sometimes it's a role reversal in Asian fireflies. And we don't exactly know what their function is. So that's another call to action. We need many more flash communication studies for Asian fireflies, and also how pheromones potentially play maybe a role in there.
And a little comment about the origin of adult bioluminescence, because the niche of nocturnal flying predators in the time when it evolved, maybe there were flying predators that we don't know about.
Yeah, we try to cover everything, fossil record, and and we couldn't come up with anything. But of course, that's always right. That's always the case. Another argument for predators not playing a role is actually if you look at the defensive chemicals themselves. So if you look at lucibufagins, only Lampyrenae produce lucibufagins. So only they have defense chemicals. And of course, they evolved as a group much later than a lot of the bioluminescence that we see today. So even if it was different predators, right, they would have been defended and they wouldn't have been able to learn that light means bad taste and a bellyache or whatever that would cause.
Yeah, but maybe there are other things than lucibufagins
That is true.
that we don't know about yet.
That's another, we need young researchers.
More studies all the time.
scientists. We've already saw that we look at bacteria, right? Molecules. So maybe we need to look more widely about different kinds of defensive chemicals.
Thank you.
Yeah. Thank you.
Thank you very much for your presentation. I'm thinking that the Central American region includes Mexico. Is that right?
Actually, yes. So one of the northernmost biogeographic region reaches all the way up
Okay.
through Central America into Mexico. Yes.
Did you have access to any information from the rest of the countries in Central America?
No, unfortunately not, but we hope that we can incorporate more of them. We have a series of Malay strap samples from Costa Rica that are closer by, but Miriam Aquino is joining our lab, who was previously worked with the Zaragoza lab, so hopefully we can figure out how to make sure that all the species richness here can be represented on the worldwide phylogeny as it deserves.
Okay. Thank you.
Just a little suggestion on how we define a species has lost its ability of bioluminescence. Because many day-flying diurnal species which are believed to having no ability to giving out light, And if you look at the specimen, you don't see any light organ, but many of them, at least in Hong Kong, almost all of them, all of the diurnal species coming from different subfamilies, if you put them in dark room and then use long exposure photography,
Mhm.
you see the light.
Mhm.
But you
Great.
don't see it by bare eye.
Mhm. So that,
So
go
yeah,
ahead. Sorry.
it may not be functional,
Mhm.
but you cannot say the ability of bioluminescence is completely lost.
Yes.
It's still there.
That I
Yeah.
think that's the same argument, right? That you can do for pheromones. Just like pheromones are not completely switched off, the bioluminescent ability is not switched off. So we know a lick, Puranas corascus, doesn't have light organs, but it has a fully functioning luciferase, right? So, and I think that speaks to what Joe Cicero talked about yesterday, that it's not an either or, it's actually degree, of how much you use it and what's the benefit of maintaining structures versus the cost of maintaining them. And so that may be really depend on individual firefly species. But I'm curious, or if you look at your fireflies, are they emitting light? Is that the cases where they emit from the whole body? Or is it from vestigial larval light organs, or is it a complete adult light organ that they somehow is cryptic and you don't see? Mhm. Okay. Mhm. Yes. Yeah, yeah, yeah. Mhm. We have an example like that in North America. It's Lucidoda atra, which is a diurnal firefly, but it has vestigial little dots of light organs, which are the larval light organs. The adults still, some of them still maintain it, and they actually glow bright green for those individuals that have it. So one of the things I didn't have time to go into, but what we're trying to do when we look at light organs, we just try to use, and whether you have flashes or glows, we try to distinguish the glows from vestigial organs from those of full-blown adult light organs. But again, that's all in process, and I hope by the end of the year we are close to getting the manuscript together to actually share it with. And yeah, I would be interested in your comments. Okay, well, thank you so much.
Thank you, Professor, Doctor, and everything. We have a couple of gifts to you.
Well, let me hand you this.
Thanks a lot for joining this event.
Thank you so much. I really appreciate it. Thank you. Okay.
Our students prepared this teaching material, and now we share with you.
Great. Thank you so much. Appreciate it. Thank you.
We have an announcement, so another surprise. There is a building, construction here, and there's a problem with the internet there. So we are trying to solve that. So we need to wait a couple of minutes in case we need to stay here for the rest of the program. But we need just the problem is the coffee break is prepared on the in the auditorium. So it's this just last minute problem. So give us 5 minutes, please. Okay, coffee break.